Hello my dear students, I am Dr. Vaishaly Bharambe and today I am going to teach you how to draw the microscopic structure of retina. So let's begin by first drawing this rectangle. We are going to divide this rectangle into 8 parts. Now you must be wondering, when retina has 10 layers, why are we dividing it only into 8 parts? Because 2 of the layers are actually just membranes. So we don't have to draw a separate section for them. Draw this division very lightly. It should only act as a guidance for you. Okay. So let's begin by drawing the first layer of retina, which is a layer of pigment cells. So draw them well. Observe how the apical region of these cells shows such microvilli-like appearance. So draw those also. The next structure we are drawing are the outer segments of rods and cones. Outer segment of rod is thin and long. That of cone is almost conical and triangular. So here observe the difference in their thickness. So these are the outer segments of rods and cones. Now we have to draw the body. So this is the body of the rod, the body of the cone. Observe how their axonal process is going further deeper into the retina. We now focus on creating the axonal region of the rod and cone individually. There you have your rods and cones drawn. Let's now draw the next cell, which is the horizontal cell. Okay. So that's the body of horizontal cell, and these are its processes which are reaching out to the rods and cones, synapsing with them. The next cell we will draw are the spindle-shaped bipolar cells. Each is having a dendritic process directed towards the rods and cones, and the axonal process directed further deeper into the retina. Let's now begin to draw the dendritic processes, making them synapse with our rods and cones. Observe how the horizontal cells are part of this synapse. Let's now draw the axonal process of these. For that I need to draw my ganglion cell. It's a large cell and its processes are directed inwards, and its axonal processes are directed further into the retina, finally meeting together to form a bunch of axons which form the optic nerve. Now let's make the bipolar cells synapse with the ganglion cells. Let's draw just one more ganglion cell to fill this space and let its axon go further inwards to participate in formation of the optic nerve. Now let's add one more cell, which is the amacrine cell. See how the processes of amacrine cell are participating in the synapse between bipolar cells and the ganglion cells. So now we are going to add a large cell called the Muller cell, which extends from the innermost portion of retina to the segments of rods and cones in this manner. These are large cells which support all cells of the retina. On the inside, these cells spread out further, separating finally the retina from the vitreous body by forming the inner limiting membrane. Let's now begin to color these cells to make us understand each cell properly. So this is my pigment cell. The light green and the dark green are the rods and cones respectively. The blue are the bipolar cells, the pink are the horizontal cells, the orange are our amacrine cells, the yellow are my ganglion cells, and finally the peach color are the Muller cells. You can color them whatever color you want because this is basically a diagrammatic representation of retina. Let's add the nuclei now to the pigment cell along with a lot of black melanin to make sure the pigment cells have ability to absorb excessive light that enters into your eyeball. Let's add the nuclei to the rods and cones, to the horizontal cells, to the bipolar cells, amacrine cells, ganglion cells, and the Mueller cells. There you have the diagrammatic representation of your retina. Let's quickly revise this. This is the pigment cell. These are the rods and cones which act as receptors for vision. When they receive visual impulse, they convert it into an impulse that is passed on to the bipolar cells, which are the first order neurons in the visual pathway. These then pass this information to the ganglion cells, which are the second order neurons in the visual pathway, which then transfer the information through their axons, which form the optic nerve, relaying this information finally into your visual cortex. Supporting all this activity are the horizontal cells, the amacrine cells, and the Mueller cells. Thus, pigment cells form the first layer of retina. The outer segments of rods and cones form the layer of outer segments of rods and cones, the second layer. The innermost edge of your Mueller cell, which is towards the pigment epithelium, is called as the outer limiting membrane, that is third layer of retina. The nuclei of rods and cones form the outer nuclear layer, fourth layer of retina. The synapse between rods and cones bipolar and horizontal cells forms the outer plexiform layer, the fifth layer of retina. The nuclei of bipolar, amacrine, horizontal and Mueller cells form the inner nuclear layer, which is the sixth layer of retina. The synapse between bipolar and ganglion cell forms the seventh layer of retina, the inner plexiform layer. The layer of ganglion cell forms the eighth layer. The optic nerve fiber layer forms the ninth layer. And finally, the innermost membrane of the Mueller cell forms the inner limiting membrane, which is the 10th layer of retina, separating the retina from the vitreous body. So this was the diagrammatic representation of retina. Let's now draw the H&E diagram. Okay, so let's pick up your H&E pencils and let's begin the diagram of retina. Let's first draw the pigment cells. Color them. Draw their nuclei. Add a good amount of pigment to them. The next layer, as we know, will be the layer of outer segments of rods and cones. Observe how the outer segments of rods are thin, whereas that of the cones is thick and almost triangular. Make sure you add both the outer segments in this layer. Now let's draw the outer limiting membrane. Let's now draw the layer of nuclei of rods and cones. Remember, these nuclei are a huge number and you have to draw a very densely packed network of nuclei here. Draw the background because every cell needs support. This becomes your layer of nuclei of rods and cones or the outer nuclear layer. Now let's begin to draw the outer plexiform layer, which is a synapse between the axons of rods and cones and the dendrites of bipolar cells and horizontal cells. So this is a fibrous layer. The next layer will be the layer of nuclei of bipolar, amacrine, horizontal and Mueller cells. But here make sure you draw fewer nuclei because the number of nuclei are way lesser than the number of nuclei found in the layer of rods and cones. This layer is the inner nuclear layer. Color the nuclei. Let's go to the next layer where the axons of the bipolar cells synapse with the dendrites of ganglion cells and amacrine cells. So again a fibrous layer. The next layer is the layer of ganglion cells. These are few cells, large in size. Make sure your diagram reflects that. Color the background. Color the cells. Draw their nuclei. And we will now focus our attention on drawing their axons, which finally form the optic nerve layer. See how their axons are emerging out, bunching together to form the layer of optic nerve fibers. Finally, let's draw the inner limiting membrane, which is the innermost layer of the Mueller's cell. This layer will separate the retina from the vitreous body. And there you have your diagram of retina. Let's understand it once more. That these are the pigment cells. You can see the melanin pigment in them. They form the first layer of retina. This is the layer containing outer segments of rods and cones, forming the second layer of retina. This is the outer limiting membrane formed by the outermost layer of Mueller's cell, forming the third layer. These are the packed nuclei of rods and cones, forming the fourth layer. This is the plexus of nerve fibers, forming the fifth layer. These are nuclei of bipolar hemicrine and horizontal and Mueller's cells, forming the sixth layer. This is again a plexus of nerve fibers, this time synapses between bipolar and ganglion cells and hemicrine cells, forming the seventh layer. These are the ganglion cells, forming the eighth layer. These are the optic nerve fibers, forming the ninth layer. And this is the inner limiting membrane, forming the 10th layer. These are the 10 layers of retina. The innermost layer separating the retina from the vitreous body. So the 10 layers, one last time, let's revise. Pigment cell layer, layer of outer segments of rods and cones, outer limiting membrane, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, layer of ganglion cells, optic nerve layer, and inner limiting membrane. Thus we complete very quickly the diagram of microscopic structure of retina. I hope you also understood the first order neuron and the second order neuron in the visual pathway. Thank you, students, and I'll see you in my next microscopy diagram video. Bye.